Hello, I am Dirkx.
And I'm Rick Helmich.
We are both researchers at the Danish Institute and Neurology Department of the Radboud University in Nijmegen. And in the next few minutes we will tell you about a new research we conducted here, where we found that dopamine controls Parkinson's tremor by inhibiting the cerebellar thalamus.
Parkinson's disease is a progressive neurodegenerative disorder characterized by bradykinesia, rigidity, and a 4 to 6 Hz resting tremor. The pathological hallmark of the disease is nigrostriatal dopamine depletion. However, in contrast to bradykinesia and rigidity, the relationship between tremor and dopamine is unclear. For example, the response of tremor to dopaminergic medication varies greatly between patients. While some patients respond very well, others show a remarkably dopamine-resistant tremor. To resolve these discrepancies, here we asked ourselves where exactly in the brain dopamine acts on the tremor circuitry. In previous work, we have identified the cerebral network involved in Parkinson's tremor using a new method based on concurrent EMG and fMRI scanning. Here you see an EMG regressor of a single patient describing tremor amplitude over time during scanning. Using this method, we have shown that Parkinson's tremor is associated with increased activity in the cerebellothalamic cortical circuit. We also searched for transient brain activity at the onset of tremor episodes. And this was associated with cerebral activity in the basal ganglia. Together, these findings suggest that Parkinson's tremor is triggered by the basal ganglia and amplified in the cerebellothalamic cortical circuit. In a subsequent study, using dynamic causal modeling, we confirmed the hypothesis that tremulous activity starts in the globus pallidus and was then propagated to the cerebellothalamic cortical circuit through the motor cortex.
In the current study, we use these same methods to test where dopamine influences the tremor circuitry. To answer this question, we studied a cohort of 15 tremor-dominant Parkinson patients. All these patients were scanned using functional magnetic resonance imaging, while simultaneously recording tremor using electromyography. Importantly, patients were scanned on 2 separate days. One day, they were instructed to stop their dopaminergic medication 24 hours prior to scan time, whereas on the other day, they continued their dopaminergic medication as usual. This allowed us to study the effect of dopamine on cerebral tremor-related activity. We found that dopamine significantly reduced tremor amplitude-related activity in the ventral intermediate nucleus of the thalamus. Furthermore, we found that dopamine significantly reduced tremor onset-related activity in the pallidum. We have now seen that dopamine acts on both the thalamus as well as the basal ganglia, but we cannot distinguish which of these areas is the primary target of dopamine. To investigate this, we used the multivariate approach, called dynamic causal modeling. This is a method where you statistically compare different cerebral tremor networks to identify a model that best explains your data. To study the effects of dopamine on the cerebral tremor network, we used our previously identified tremor network consisting of basal ganglia and the cerebellothalamo-cortical circuit. Specifically, we compared 17 models where each connection of this tremor network was modulated by dopamine and one where there was no influence of dopamine. Here we found that the model where dopamine influenced the self-connection of the thalamus best explained our data. Specifically, the connection strength of the self-connection increased when on dopamine, meaning that dopamine increased the self-inhibition of the thalamus. This fits with our finding that dopamine reduces tremor-related activity in the thalamus. The benefit of the multivariate approach, however, is that this model comparison shows that the effect of dopamine on the VIM is not mediated through the basal ganglia. Importantly, the modeled effect of dopamine by DCM correlated with the clinical dopaminergic response of tremor and tremor only. In other words, thalamic inhibition is greater in patients with a relatively dopamine-responsive tremor. So in conclusion, this research provides several new findings. First, we found that dopamine controls Parkinson's tremor by acting on the cerebellar thalamus and on the pallidum. This finding suggests that the degeneration of dopaminergic projections to the VIM and to the pallidum have a role in the pathophysiology of Parkinson's tremor.
And second, we found that these effects can explain clinical variability. patients with a relatively dopamine-responsive tremor had a larger influence of dopamine on the VIM than patients with a relatively dopamine-resistant tremor. And this indicates that thalamic self-inhibition might be impaired in patients who have a dopamine-resistant tremor. Thank you for your attention.
Thank you.